Okay, I made some changes to our schedule. I know some of you had walked in and said, are these notes, are these worksheets, what are they? Well, if we remember, the papers that you were given did not line up very well with the presentation that we had for you. So we skipped over quite a bit, like in section 12-2. I mean, it says 10, should say 12. I got that correction made on this presentation. Some other changes. I will not be here tomorrow. Mr. Wagner will. We'll have a paper for you to work on that's due on Friday. Your vocabulary quiz is also on Friday. So with that, okay, how many people will be gone on Thursday? 1, 2. Okay, so what that means is, repeat after me. Even though we're not here on Thursday, the vocab quiz is on Friday. You tell me that again.
Even though we're not here on Thursday, the vocab quiz is on Friday.
Okay, so when is your vocab quiz? It's on Friday. Is there any question over that? Okay, so I understand there are possibly some definitions that are in your vocabulary that, see, this is where later down the line, I don't get what we're talking about because you're talking the same time I am. So I think a general rule of thumb is probably something you learned in elementary is when I'm talking, you're not. Okay? So I understand there's probably some vocab terms that are possibly not in the back of your book. That's why we set due dates and we can discuss that. I'm going to hit pause for just a moment so I can see when it is we had them due. Okay. We just said that they were due tomorrow, but with that, let's get on the same page and have them due. Thursday's not a very good day, so Wednesday. So what I want you to do, how many of you have done as many as you possibly can? Okay, so with that, simple question about how many of those papers that you have now in front of you that you don't have a definition for?
2, 4, 6, 7.
So, okay, so just start telling me some of those terms so we can address that. No, I just want to know what the terms are.
Nucleotide structure.
Okay, nucleotide structure. Okay, next. Purines, and then probably pyrimidines. Okay. Okay.
Duplication. Delegation.
That should be it. Maybe one more. 6, 7, 8. I thought I heard 9. That's 8. Oddly enough, some of these, and we'll cover them a little further in depth tomorrow, but as I look at this, I see 1, 2 of them that we did talk about, and that would be these 2. Okay? So when we put that to practical terms, what do you think DNA helicase is?
It's the enzyme group that splits up the DNA.
Okay? So we're not going to go too far in depth with this. Remember, you had some of these nucleotides in here, and okay, we'll just use that as an example. So how do you think that DNA molecule is held together? It's a type of bond. So these are what we call hydrogen bonds. Okay? Then in our diagram, we put like a circle here, and it illustrated that there was an enzyme moving in this direction. What enzyme do you think that would be? And you can just say helicase. Okay? And that's not the only enzyme we talked about. What might be another enzyme that we discussed? No.
The one that starts with P.
Now, the reason that we can say just helicase is there's only one type of helicase enzyme, and that is DNA helicase. That is it. Okay? When it comes to polymerase, what is everything that you can tell me about polymerase? There must be more than one that exists.
So the synthesis of new DNA molecules.
Okay? So that must mean we have DNA polymerase, which we also have what? RNA polymerase. Okay? And again, we'll go a little further in depth when we get to that. And just so when you're looking at this, let's not make this really, really extremely difficult. We'll try to keep this as simple as possible. What do you think if I go to the copier and I put this in here, I hit start, all of a sudden it goes through the machine, what spits out the other side? Another copy of it. So is that the same as what we see here? Yeah. So with that, a copy is the same thing as what you could say is a duplicate. Okay? So that's what happens in DNA. Again, there's a little more to the terminology we want to get to, but we just want to say it's not going to be rocket science. Okay? So if that's what happens when you have a duplicate, that means you have two of the same molecules. That's not good. Okay? In other words, you're having the same thing happen. You have the same that's on the inside, same that's on the inside, same that's on the outside, same that's on the outside. Yes?
In DNA, can A ever be with U?
No. Correct. Okay? Then what would you think if, see, I'm just trying to, this was long before your time. They were making fun of the grounds crew in Kansas City for football. So this is what it was supposed to say in the enzyme or in the end zone. But after many, many hours of work, for any of you who have ever seen our grounds crew paint the football field, it's not like you grab the machine and an hour later you're done. How many of you believe that? Good. I'm glad that I didn't see any hands go up. But how do you suppose that would be different when you have to paint an end zone?
A lot more paint.
Why is there going to be a lot more paint?
Because the letters got to be molded.
It's all solid color usually. So if you're looking at the end zone for the Chiefs, okay, here's the end zone. And it's all painted. What color do you think?
Red.
It's all red. And then what do you suppose this is supposed to be?
White.
Really? Yellow?
Ow!
It's white.
And what are we doing other than leaning back?
Sorry.
And what are you thumping her in the head for? Okay. So when this is what it's supposed to say, look at what that says, and what do you think from just very little discussion, what do you suppose that might mean? Okay? Yeah. So the commercial goes on, one of the linemen comes up and he looks at this, and of course these are probably all capitals, they should be. He looks at this and he says, "Man, that looks good, but who are the chefs?" Okay? So what had happened is after many, many, many hours, I'm just going to say 3, I guess that's not many, this is what it's supposed to say. This is what the final product was. So what happens is a mutation is somewhere. You could look at these and maybe for some odd reason, there's no explanation for it, probably not on the inside, but probably more so on the outside when this replicates. We know this is G, this should be A, and then that's supposed to say a T, but there's nothing there. It got deleted. That's what, it's a type of mutation where a nucleotide for some reason or another just disappears. That's a deletion. Okay, so we went over quite a bit of an amount of time, or a lot of material in a short amount of time. Then the last thing I want to do is just very quickly show you what I think this one is adenine and guanine, this is cytosine and thymine, I think. And we're not talking about those that bond together. That's not what we're referring to. But rather, I'll give you the page number, and then you can see, oh, well, that makes more sense. And that is page 329. Okay? The reason I say that is up here on top, it has what we call a purine base and a pyrimidine. It tells you what the difference is. Okay? And that's all you're doing. Page 329. Okay. So what we want to do next is try to get on the same page. We've applied all this material, but again, what you had in front of you just did not match up with what was on here.
Will we ever finish the video about the surgeon?
We did not. That is still on today's schedule. I just don't know how far we're going to get. Okay, so some of this, just follow along. I took everything that was on here and I just copied it into Word, okay? A lot of this hopefully makes sense, okay? In other words, what did we talk about here? No surprise. What does this helicase do?
Splits up hydrogen bonds.
Okay, so that's no surprise. That's what we saw right here. Here's our helicase enzyme, breaks those hydrogen bonds. Okay?
So DNA helicase splits.
Yep. It's the enzyme
Hydrogen
that
bond?
splits
On DNA.
It is.
Okay? So where is the only place that you find DNA? That's different than RNA.
it up.
Why are you right? Well, one thing we could presume here, where are these nucleotides floating freely? Certainly not in the cytoplasm. I mean, those are, but not for this process, okay? Because when we find these nucleotides in the cytoplasm, they come in 3-letter groupings, okay? They either come in a grouping called a codon or an anticodon, but we'll get to that at a later time, okay? So then we talked about DNA polymerase as well, okay? So that causes hydrogen bonding to occur and seal that strand back together in a double helix. So what happens is rather than just having one DNA molecule, you end up with 2, okay? So that's what we're trying to illustrate here, okay? So with 1, you get 2 of those, okay? All right. What did we say? What does it mean to be a scribe?
To read and write.
Yeah, you could say middle class. That's not the explanation I was looking for. You're not wrong. It's just applied incorrectly. Okay? So when we see this transcription, what is it that is reading the DNA? True, but in this case, it's another type of strand of nucleic acids.
I know what it is. Nucleotides?
No.
Oh.
Messenger RNA?
Messenger RNA is the scribe because we said that is either Wyatt or Jackson, depending upon who is taking the message from the sideline to the huddle or the ribosome. Okay? So this is your coach. This is your players. They are your readers or your scribes because they are reading the directions from the coach. Okay. And then this is just we get further into depth with, this is the reasoning for that replication, okay? Now, this messenger RNA directs. So here's Jackson or Wyatt. They direct the assembly of the players or the proteins at the ribosome, okay? Because you could say that the amino acids are your separate players, okay? Whether it's a center, a guard, I almost said tackle, that's not right. Center, a guard, an end, a receiver, and then maybe, a running back? I don't know. Fullback, whichever. So whatever combination, get to add to 10. Nobody, there's only one person that caught that.
I heard something you had.
Say that again. There's only supposed to be 9. So that must have been a duplication. A mutation known as a duplication.
You got you there.
Okay? Right. So you could say, yes, that is a penalty. And that happens in the body. That can be a really big penalty.
It's been like 3 times.
Well, it was.
Buddy has the most fouls on our team, yet he plays the least.
He has the most talent.
Okay. Does everyone have this content that wants it? Okay? So in the most shortest definition we could give about this process, okay, is this right here. Okay? Because how we made this analogy, where is DNA? Now, our analogy, not the cell. Our analogy when we looked at the football field. Where is this? True. The sideline. Because that's where your coaches are. They've got the instructions. Okay? Then, of course, there's a little more to this, but the formation of these proteins, it's encoded on the DNA that the coaches have. Yes?
What would be the transfer RNA, like in your?
That's way down the road.
That's Mr. Jack.
Okay? So, again, here's your side lane. How does this information get from here out to the ribosome or the huddle?
By the messenger RNA.
The messenger RNA reads that. And then that information tells not just the amino acids, but the players where they need to be assembled at the huddle. Okay? So the reason it's called protein synthesis is this information directs in a coded message how those amino acids have to be assembled. Okay, now does everyone have this? Okay, I want to do this one and get down to here. Okay. So is this a structural difference as opposed to DNA? This sugar here. No, it is not, because they both have ribose. Okay, that's why it's deoxyribose and then ribose. If you take biochemistry from here, you actually are responsible for drawing the structural differences between those for DNA and RNA. But leave that where it's at. That's a senior level application. Now, can we find uracil in the nucleus?
No.
That's a trick question.
Nope. Nucleus is the uracil.
Because that's where, because DNA is held in the nucleus.
If there was no uracil in the nucleus, how could it get out to the ribosome? The reason we say it's a trick question is, yes, this is found in the nucleus.
How? It's just floating around in there.
Mm-hmm.
Yep.
Then how come the DNA doesn't pick it up and the RNA does?
Isn't the transfer
I suppose it's because of the enzyme activity of RNA polymerase. That's the best explanation I've got. It's because of that enzyme action.
Science is kind of stupid, honestly, if you think about it.
Mm,
It just doesn't make a lot of sense.
it's just because it's new. And if we tried to go into the depth that would really explain that, that's way too high. Way too high. Just accept it that it's found in the nucleus. Okay? And we've addressed questions like that's still a good question. Don't say, "Oh my God, I asked a bad question." Don't do that. Okay? The reason I say just accept that. Okay? What I mean by that is if we want to find the circumference of that shape, you have to accept that you have to multiply by what number? Yes, so you just accept that because it's something you know you have to do. Okay,
Yes.
so what we want to talk about is, do we remember the young boy named Wasim? Okay, that's what we call an encephalocele. Okay, now, why did his nose look so big?
Because his brain was leaking out of something that was missing a part of it was leaking out of his nose.
There was a structure in the top four.
Did you get that?
No, I heard a couple mumble jumbles.
Okay, so what happens is that everything that you could cover about that's not going to make any sense because it's something that you can't see, but something that you can see and have experienced. How many of you have looked after, I'm not saying just younger brothers and or sisters, I'm talking about infants or do babysitting of younger infants, okay? So what we mean by that is as follows. If you are holding an infant and if you are just curious for some odd reason, you've heard about these before, you feel around their cranial area, okay? So what happens is there's a process called ossification. And what that means is those very same soft spots that you're talking about is cartilage, okay? But it doesn't remain cartilage because if it did, we would have those same soft spots, but we don't. You can press on your head and you're not going to go into your cranial cavity, okay? So in this case, somewhere in the genetic code, maybe there was a deletion, maybe there was a duplication, maybe there was an insertion. Any one of those mutations was severe enough that caused the cartilage that was inside the base of his skull, it just never fully ossified. So it just remained as cartilage. So what had happened is his brain just continued to do, like you said, what? Leaked into his nasal cavity. Now, the idea behind that brain tissue is you don't want to have any type of brain tissue leak into a cranial cavity. Yes?
So, does that mean then, like, some of his skull was hollowing out then? If it was just leaking?
I thought maybe I didn't do a good job of explaining that. Okay? We said with all these soft spots, for some reason in his nasal cavity where that was still cartilage, it just did not form to solid bone. So when that happened, that's what allowed that, if you want to call it brain tissue, to leak into his nose. But it's non-functioning. Okay? I mean, that doesn't say, oh, it's non-functioning, so it's not that big of a deal. No, of course we're not saying that.
No, I
If he
still,
hits
I
it too
got
hard,
that.
isn't he gonna die or something?
It's exactly, it's just going to keep on leaking unless that is fixed. Okay, so we will pick up with that at this time. So this really, we talked about enough of what we need, because I'm afraid if I go any further, we're not going to be able to see this on the Science Channel. So keep in mind those schedule changes. Worksheet tomorrow, vocab quiz on Friday.
Facts.
Okay. And your
Facts.
vocabulary we will address on Wednesday. Okay? Wednesday is when these are due.
Wednesday.
Wednesday.
Yeah, don't forget
Okay.
Wednesday.
All right. We'll catch up to you next time.